I think we're going to begin. I want to welcome you all to the third installment of the BERD Clinical Trials series. And we are going to have Dr. Greg Wilding back again, which is lovely. And he's going to talk about randomization techniques. And there is a slight change in the schedule with Austin Glick coming up next. Mike Lamont will actually be presenting next week with Austin with his second section. So, Austin, thank you for your flexibility. I appreciate it. And I'm going to make you co-host in a moment because you are coming on a little later and I'm going to turn it over to Dr. Wilding. Thank you.
Thanks, Catherine. Hi, everyone. I'm Greg Wilding, for those that I don't know. I'm a professor in the Department of Biostatistics, and I also direct 2 units on campus, which might help you with your trial needs, potentially, and I'll talk about those a little bit later. Let me just kind of get right into the talk, and why am I even doing this particular talk for our clinical trial series? Well, if I want to establish causation of some kind of intervention, right? So I have an intervention, I want to establish the cause and effect. I can do that a number of ways. The gold standard is to do a randomized experiment. And so that's why I really think that really knowledge of randomization, the reason why we randomize and how we randomize, I think that's really required knowledge for our scientists. And so you might say, well, Greg, why do you need an entire talk on it? Why are you babbling on for 50 minutes about randomization? Because isn't it a coin flip? Not literally, but figuratively. And it is, and it can be, but that's often not the best strategy. And so we're going to consider a bunch of different algorithms. All these algorithms have this goal of comparability with the idea that I want to establish statistical validity. I want to have a very efficient statistical procedure in play with my clinical trial. And I also want to ensure some credibility. And I'm going to do that through obtaining different forms of balance. And so that takes us to our learning objectives today. You should be able to understand the rationale behind a randomized experiment. You should be able to describe some really common utilized algorithms and understand their pros and cons. And then I'll show you how to generate some randomization lists in Excel. I'm not an Excel user. I know a lot of people on the call are. One thing to note with a lot of the randomization procedures here, there's a lot of thick mathematics behind it. I won't torture you with that today. We're going to keep it very applied like we do all of the workshops for this series. So when I think about establishing causation, right? That usually involves allocating individuals to some groups, right? So I get a recruited sample. I have some patients or volunteers that meet my eligibility criteria, and then from there, there's some kind of assignment process where I might assign them to, let's say, 2 groups, right? It could be more, but let's keep it simple here. And then since it's a trial, I'm going to follow them forward in time. It's a prospective experiment, and at some point, I'm going to do some formal comparison of outcomes. And so that brings up the question, how do I actually make my groups comparable? And when I say comparable, I mean homogeneous in terms of sample size and or important variables that I might measure at baseline, these variables being prognostic of outcome. And so I could do that non-randomly, right? That would be easy enough. That I established some criteria. If you meet criteria 1, you go to group A. If I meet criteria 2, you go to group B. And if I do that, and especially if I have an unblinded clinical trial, because a lot of our clinical trials have to be unblinded, then when I go to analyze the data, my observed treatment effect in the data could be a result of the actual effect in the population. And that makes sense, because why else would I do the trial if I don't think that's at least a possibility that I have a non-zero effect? It could be due to chance, and that's something we have to accept if we're going to try to make conclusions based on data. I can make things like type 1 errors, as they're known. It could also be due to confounding variables, and these would be variables that are related to my outcome that might be distributed differently at baseline, and I could also have differences in handling and evaluation during the study. Now, reasons 3 and 4 are really problematic because that can lead to biases when I go to assess the treatment effect. This has been well documented and studied and discussed in the literature. I could have assignment bias, I could have assessment bias, I could have selection biases. There's all sorts of biases that I can worry about. And so if I have a non-randomized trial, that's going to be a real challenge in terms of making causal inference. And I can get at causal inference with a non-randomized trial. The problem is I have to make a number of assumptions, and these assumptions are going to be very challengeable. And so that creates some problems for me. And so really, I'm better off dealing with a blinded randomized trial, because what's going to happen is, of course, at the end of the trial, my observed treatment effect might be an effect of one, my effect of my intervention. It could be due to chance, but I'm eliminating reasons 3 and 4. And so that's the reason we go through the effort of doing a randomized trial, is because then I can get at causal inference, and I can do that in a very simple, almost assumption-free way. Now, there are also some statistical benefits to a randomized experiment. You're obviously used to this idea of a model-based inference procedure, like a t-test would be a famous example of that. There are a number of design-based inferences that are tied directly to randomization. That's really beyond the scope of today's talk, but just be aware there are some additional advantages that we won't discuss here. Now, of course, there's some disadvantages with a randomized experiment. There's ethical issues. You may be dealing with patients or physicians that might not want to participate in your trial. You have this idea of volunteer bias. Generally, people that volunteer for trials are different than those that do not. And so that creates a bias when I think about generalizing to my population. And then there's a lot of issues around interfering with the patient-doctor relationship, because I am randomizing the treatment to the individuals. So what we're going to do today is we're going to review some of these randomization procedures. There's really hundreds and hundreds, maybe even thousands of algorithms out there. What we're going to do is we're going to focus only on a few to keep this fairly concise. I'm going to focus on algorithms that are commonly used and accepted by the scientific community, fairly easy to implement. They fall into these 4 classes here. We have complete randomization. We have what's called restricted randomization, aka treatment adaptive randomization, covariate adaptive, and response adaptive randomization. So these last 3, these are adaptive in the sense that the probability of being allocated to a given treatment group depends on prior information. A lot of you would be familiar on the call about with at least the term adaptive designs. Sometimes people will say that if you use these sort of randomization algorithms, it's an adaptive design. Some people say that's not enough, just so you are aware of that. Now, in terms of the rest of the talk, I'm going to be focused on 2 groups, even though most of what I'm going to talk about could be extended to any number of groups. Generically, I'm going to use the term treatment and control, which is what we generally do when we talk about clinical trial methodology, even though I might not necessarily be applying a treatment in the sense of something that will help disease. Or I might talk about groups A and B. And then we see N that corresponds to the total sample size, N1 and N2, that'd be the sample size for my 2 groups. So beginning with the simplest form of randomization, this is complete randomization. It's also known as simple randomization. It's essentially your coin flip. You're going to have this known consistent probability being allocated to one of my 2 treatment groups during the trial, and that will not change at any point in time. Now, to implement it, why it's so commonly used, it's really easy to implement. All I have to do is come up with a source of random numbers. And I'm going to say they're really pseudo-random numbers. Usually where we get these from is a software package where there's an algorithm in the background that creates these numbers that are random enough. But usually they're based on some kind of algorithm that might be based on the time of your computer or something like that. Specifically, I'm going to use uniform random numbers. And these work out nicely for generating randomization lists. A uniform random number is a number between 0 and 1, where the probability of a number between any two lengths is the same. And so what I mean by that is the probability that my uniform random number is between 0.2 and 0.4. Well, the difference between those 2 numbers is 0.2. That's also the probability of that happening. And that will be the same as the probability of observing a number between 0.63 and 0.83, again, because the difference is 0.2. So if I can generate these uniform random numbers and I come up with an assignment rule like you see here in the box, that's all I need to do to come up with to generate a randomization list. So let's say that if my uniform number is between 0 and 0.5, well, then you're assigned to group A. If it's between 0.5 and 1, then you're assigned to group B. So what I'm going to do is I'm going to show you how to do this in Excel. I personally don't use Excel. I use a statistical software for this, but I know a lot of Excel users are on the call. If you're going to use Excel, you just need to utilize these 2 functions, the RAND function and the if function in Excel. The RAND function generates a uniform random number, just like I talked about in the previous slide. The if function specifies an if then else statement. If you're not familiar with that, essentially, you're going to have a condition. which is the first argument. If the condition is true, then it's going to return the second argument. If it's false, it's going to return the third argument. And so if I'm going to generate a randomization list using complete randomization, all I have to do is just open up an Excel workbook. What I've done there is in the first column, I've created a subject number, whatever format you want is fine. Next, I use that rand function and just fill in the cells with randomly generated uniform random numbers, right? Again, numbers between 0 and 1. And then what I'm going to do in the third column, I'm going to apply that if function. And so if the number in the second column is less than 0.5, then that subject gets an A. If it's greater than 0.5, they get a B. And so I can go ahead and just fill it in, and boom, you're done. You have your randomization list, right? Pretty straightforward. One thing I do want to caution you about, you are using Excel, and this is one of the drawbacks of using something like Excel for science, right, and for analysis, is that every time I open up that Excel workbook, it's going to redo those numbers. It's just an artifact of that random. So what I would recommend you do is when you first generate that randomization column with the assignments of A and B, copy and paste not the formulas, but the values. That way, if you open up that spreadsheet again, those values, those assignments won't change. Now, if it did change, with complete randomization, it's actually not that big of a deal from a scientific perspective. But if you get audited, that could be an issue. So if you're dealing with a funny mechanism where there could be an audit and they want to know exactly how did you assign subjects to your different treatment groups, well, if you open up your Excel workbook and it's a different list, okay, that's a bit awkward, right? So you want to make sure that you document that randomization list. And so just be careful with that. Now, the advantages of complete randomization is it's super simple, right? It took me like 2 seconds to generate that list. And also, it's impossible, as long as you don't make that list public, it's impossible for someone to guess what the next assignment is. And I point that out because with some of the procedures we're going to talk about later on, that's not true. So I can go ahead and use complete randomization. Another drawback is, and this is why I personally don't use this a lot, especially with smaller trials, is that you can end up with a bit of an imbalance, right? And one of those imbalances is going to be terms of sample size. And so this is actually something I can calculate. And your intuition might have already told you this is potentially a problem, right? Because we're all very familiar with the idea of a coin flip. When you first learn probability, they usually kind of start with the idea of a coin flip because you have this known probability of a heads, 1/2. And you could talk about, the number, what's the probability of having 8 out of 10 heads, right? You can calculate that using a binomial distribution. You know that if you flip a penny a million times, right, you're going to have very close to 50-50 in terms of heads and tails. If you flip that same penny 10 times, you have a much greater chance for an imbalance. What's going on here with randomization is the exact same mathematical problem. So I can go ahead and actually calculate these probabilities. This graph you see here at the right, on the x-axis, you have the percent in one of my groups. And then the y-axis, you have the probability of such an imbalance or greater. And so if I have a really small trial of, say, 10 people, and this would be more something like you might have with a pilot clinical trial where you're assessing feasibility. If I want to calculate, if I want to see what the probability is of having an imbalance of 70% or more, so that means 7, 3, 8 or 2, or 9 and 1, or 10 or 0 in both directions, right? The probability of that happening is about 1/3. Right? So that's a substantial probability. Now, if I increase the sample size, of course, that probability will go down, right? And so that's the beauty of large sample size. But many of us on the call are not dealing with very large sample sizes, depending on your science. And so that can create a little bit of an issue. Now, what are the issues with it? Because you might say, well, who cares? So what? You end up with an imbalance. That's what happens sometimes. Well, there's a couple issues. One's non-statistical and one is statistical. The non-statistical aspect is, I think, credibility. So when I design a study, I'm thinking about the research product I'm going to create and how it's going to be perceived by the scientific community. If someone sees that I have this large imbalance, not that I did anything wrong, but by chance I ended up with this pretty large imbalance, if they're not exactly statistically savvy, they might start to question, okay, how did they screw that up, right? And then if I can't trust their randomization, how can I trust any of their results? And so that's something I need to be aware of as a scientist, that how my results are perceived by the scientific community. Perhaps a bigger issue for me from a statistical perspective is the loss in efficiency. So generally speaking, a lot of our statistical procedures perform best under a balanced sample size. And when I calculate like sample size, when I'm planning my trials, often we'll assume that we have a balanced sample size. If in reality I have an unbalanced sample size, I'm going to take a hit in power, right? My confidence intervals are going to be wider than I was thinking they were going to be. Now, the good news is that power loss is often not great, right? Maybe instead of 80% power, I have 76% power. So it's not the end of the world. But really, I want to be maximizing my efficiency when I'm analyzing my data. And so for that reason, I'm going to want to consider other randomization procedures. And that gets us into the idea of restricted randomization procedures, where what I'm trying to do is either eliminate or reduce this sort of imbalance that can happen with the randomization. Like complete randomization, it has the advantage that I can generate this list in advance, and I'll show you on the next slide how to do that. Specifically, what we're going to look at here is something called random allocation. Now, with random allocation, the goal here is to have a perfectly balanced sample size at the end of the trial. And to really understand this, you want to think about this in terms of a classic marble urn problem. Often when you're learning about probability, you do that in the context of an urn, right? You might remember that from your first stat class, because that's something that we can visualize in front of us. So imagine an urn where you have where half of the marbles in the urn are red and half are blue. If I want to create a random sequence of red and blue marbles, I can reach in the urn, observe the color and note it. Do not return that marble and keep going through that process one at a time till the urn is empty. And I'm going to wind up with a random sequence of red and blue marbles. What I can do is apply that same idea for randomization. Instead of having red and blue marbles, I have treatment A and treatment B marbles. And that's the idea of urn randomization, also often referred to as Laughlin's urn randomization. And this is something that is fairly commonly used. How would I do that in Excel? There's a bunch of ways, but this is, I think, is a pretty simple way of doing it. Let's say that I want to randomize 12 subjects, and I'm picking 12 just because it's going to fit on the slide here. I want to allocate them equally, so I have 6 and 6 in my two groups. What I'm going to do is I'm going to have a column again for subject number, and then the first 6 I'm going to make A, and the second 6 I'm going to make B. Okay, so that's not my list yet, right? What I need to do is mix up those A's and B's. Now, how could I do that? Well, one way I could do that is to generate a uniform random number, and then I'm going to sort by that number to do the mixing up. So what I'm going to do is I'm going to highlight the last two columns, the second and third column, and then I'm going to use the sort button, right? And I'm going to sort by that third column. And that's essentially going to mix up my A's and B's. So again, really simple. I wind up with a randomization list where I have an equal number of A's and B's. Now, my talk's not done yet, right? We still got some more time. So that must mean that there's some issues with this. Okay, well, one disadvantage is that, sure, I'm going to have perfect balance at the end. The issue is that I'm going to have potential imbalances during the trial. And so that's one disadvantage. I'm actually kind of going a little bit out of order here. I kind of skipped to the second one. I'll come to the first point in a second. Um, I'm going to have balance at the end, but not during the trial. Where might that be a possible issue? That could be a possible issue if my subject population is changing over time. That's more of an issue for, like, I think, longer term trials. Um, a bigger issue, I think, with a lot of the surgeons I work with is that often your intervention might change over time. So if it's a pill, yeah, that's not going to change so much. But if it's a surgical technique, that's your experimental intervention, you're going to get better. If you're a surgeon, you're going to get better with that technique over time. And so your intervention is changing. And so if you have kind of an imbalance during the trial, that's going to possibly create issues for you when you're assessing the treatment effect. And then a lot of our trials often don't go to the end, right? A trial will shut down early if you run out of time, you run out of money, or if something has changed in the population such that you no longer have equipoise, and then you can end up with an unbalanced trial because you stopped early. The first point that I picked here is that you also, especially in an unblinded trial or a blinded trial where there's some risk of unblinding, is you have the possibility of selection bias. And so why might that come up? Well, if I know that you're going to randomize 12 people and I see by the time you get to patient number 10, you've used all your A's, I know the rest of them are B's. And so that's going to introduce the possibility of biases creeping into your trial. And that's something that we always have to be very cautious of. So as an alternative to random allocation, right, if I'm worried about this imbalance during the trial, I could use something like block randomization instead. Now with block randomization, this has this nice feature. At the very end, I'm going to have perfect balance. And then during the trial, I'm going to have at least approximate balance. And so the basic idea is I'm going to take my subject allocation slots and I'm going to divide them into capital M blocks of size small m. So my sample size is going to be big M times small m. And then what I'm going to do is for each block, I'm going to do random allocation. So I'm going to have equal number of A's and B's, and then I'm going to do this for multiple blocks, and that will comprise my randomization list. And so to really understand this, let's look at an example. So if I want to randomize my 12 individuals, what I'm going to do is I'm going to randomly come up with a block, a sequence of A's and B's, and that will be my first block. And that will cover subjects 1 through 4. I'll then do it again for the second block and then the third block, and that will create my list for my Bermuda block randomization. And so you can see by the time I get to the end here, I'm going to have an equal number of A's and B's. After every block is done, I'm going to have an equal number of A's and B's. And at the very most during this trial, I'm going to have an imbalance of 2, right? That's the mathematically, that's the worst the imbalance can be. To implement this in Excel, again, not too bad. So I'm going to create a subject column. I'm going to create a block column. For each block, I'm using blocks of size 4 here. I'm going to have 2 A's and 2 B's. You can see that you have the same sequence here. And then what I'm going to do is I'm going to create this random number here in the 4th column. And that's for the sake of mixing up the A's and B's. Now, one thing I got to be a little bit careful of when I do this sorting is I do not want to sort by D alone, because that will mix up the A's and B's across the blocks. I want to mix up the A's and B's within the block. So I am going to first sort by block, and then after block, I'm going to sort by the random number. So it's important that you add that extra level when you're sorting, and that will, again, create your randomization list that you need. And this is something that I actually use a lot. This has a lot of great properties to it. It's really simple. With a lot of these more complicated algorithms, you could screw up pretty easily if you're not paying attention. I like block randomization for that reason, that it's really simple. It's hard to mess up, which is a good property for a trial technique, since there's so many complicated things going on here. Now, there are some concerns that you got to be aware of here. Now, one is that if you're dealing again with an unblinded trial, you can guess the sequence or actually know what the next assignment is. Here's a really simple example. Let's say after I've enrolled 7 people, if it's an unblinded trial, I see that, okay, the first block was A, B, and then the second block was B, A, B. I know the next one's an A. And so you run the risk again of some kind of bias creeping into your trial potentially. And at least you have to be aware of it, and you have to take actions to counteract that. Now, how would I deal with that? Well, one thing you could do is just not reveal the blocking mechanism. I'll do that in protocols. Like if I'm generating the randomization lists, I'm often the only one that knows the block size. Or what you could do is you could use random block sizes. And so you can imagine how this would work is rather than consistently using blocks of size 4, I'm going to randomly pick a block size, say either 2 or 4, which I pick with probably 50%. And then for those blocks, I do the random allocation. And so that is going to kind of decrease your chance of guessing, of having somebody able to guess what that next assignment is. So with all these procedures, there's a lot of other restricted randomization procedures that could be used. I'll just kind of review these briefly. There's something called bias coin randomization. With bias coin randomization, it basically is very similar to complete randomization, except your probability changes when there's an imbalance. And so what I mean by that, Well, if you have a balanced sample size, well, then the next patient's going to be allocated A with a probability of 1/2. If I've been randomizing more people to B, right, what I want to do is I want to randomize to A, not with a probability of 1/2, but with a probability higher than 1/2, say 2/3 or 3/4. If I've been randomized into A more often, well, then I'm going to do the opposite, right? And so that's the idea of a bias coin. When people say a bias coin, that's usually what they mean is a coin that doesn't have a probability of half-half for heads and tails. Um, so that's bias coin randomization. There's also something called Ways earn randomization. Often people will say earn randomization, and the problem is that there's lots of earned randomizations, as you can imagine. Often what they mean is Ways earn randomization. And the idea of this is that I'm going to take my hypothetical urn that's in front of me, and I'm going to put lambda marbles of A and lambda marbles of B. And then what I'm going to do is I'm going to pick a marble out. I'm going to observe whether it's an A or B, and that's going to determine the assignment of the patient. And then I'm going to put that marble back. And if it was an A, I'm going to put a handful of size beta of B marbles in there. And so what is that going to do? It's going to increase the chance that I pick B next time. So you're, in some sense, correcting the probability as you go along. If I start to get this imbalance, well, I'm going to start randomizing it to the other group a little more aggressively, you could say, with a higher probability. There's several modifications of this, but again, these are our commonly used procedures. And these all have the advantage, even though I kind of am talking about this hypothetical urn where I'm pulling out marbles as patients walk through the door. This is a list. You can use these concepts to generate your list ahead of time. And generally, you should do that when you're implementing a trial. Now, with all these procedures, one thing that I haven't been worried about is what if I have a difference in terms of important covariates? Often we're very concerned with that at the end of the trial, I want to have groups that are pretty well balanced in terms of important factors. And what do I mean by important factors? Factors that are highly related to my outcome of interest. Generally, age often falls in that category, sex falls in that category, sometimes race. If I'm dealing with patient subjects, the stage of disease can often be a predictor. Often site can be a predictor of how well someone does in terms of outcomes. Now, the good news with big clinical trials, and this is why we love big clinical trials, is that those imbalances are going to be very rare, right? So in terms of the variables I measure, the variables I don't measure, or even the variables I don't know that exist, I'm going to have balance. Again, the beauty of big randomized trials. Unfortunately, I don't only get to work on big trials. I work on a lot of small trials. So, a lot of pilot trials, trials of more moderate sample size. It's with those trials you can end up with an imbalance. So imagine you have this study sample of these individuals you recruited, and then you randomize them, and then by chance you didn't do anything wrong, but by chance you end up with this sort of imbalance. So let the color of the figures here represent maybe some important variable related to outcome. Maybe it's age group. Maybe imagine there's 4 age groups. With some of these age groups, you'll have balance, right? You can see that I have 3 blue here and 3 blue here, but with red I have 5 and 1. And so something like this would not be that unlikely to see in your study data. And this is something that's been well studied in the literature. Um, in this 99 paper, what they did is they just did some calculations that if you have a single binary covariate with a prevalence of 30%, and say you consider a significant imbalance of 10% or more, well, that can happen with really non-trivial probability. If I consider a sample size of 30, right, that's going to happen 43% of the time. And you might say, well, Greg, that's a problem that's going to go away. And you're right. That is going to go away as my sample size gets larger. It might not necessarily go away at the rate you would like. If I go to 100, right, and that's a pretty big sample size for some of us on the call here, that still happens over a quarter of the time. And keep in mind that this is only with a single variable, right? Imagine you have sets of variables, right? It's going to increase the chance of you seeing a pretty serious imbalance. That imbalance is going to confound your treatment effect. And what do I mean by that? Well, imagine a trial where your outcome is survival. Say you have patients with really bad disease, you randomize them to two groups, you follow them forward in time, you see the treatment group has better survival, and that's great news. But then you look at the baseline information, and you see that by chance, the people in the treatment group were much, much younger and much healthier in terms of maybe other variables, as measured by other variables. That's gonna threaten the credibility of your trial. Now, when I say that, I do want to point out that that doesn't mean that your testing procedure is invalid. It's not that whatever statistical tests you use doesn't work anymore. But keep in mind when we talk about our testing procedure, that's in the case of implementing them an infinite number of times. When I talk about the properties of the t-test, right? That's if I was to perform my experiment an infinite number of times, it will reject alpha percent of the time. So in terms of my properties, that's not the issue, right? The issue is a loss in efficiency and perhaps, again, a possible jeopardized credibility. If people see that my groups are very unbalanced in terms of age at baseline, are they going to possibly question whether my trial is telling us anything at all? And so what I need to do is I need to consider something like stratified randomization, where I first, before randomizing my subjects, I define homogeneous groups. So I take my study sample, and rather than randomize right away, I observe what stratification factor level that they fit in, and then after that, I randomize to my groups to obtain a balanced sample. Now, if I'm going to do that, there's three steps here. I'm going to first identify the covariates. I'm then, for every cross classification, generate a list. And then as subjects enroll, observe what lists I use. So for the first point, how do I identify these important covariates? This is something you got to spend a decent amount of time thinking about during the planning stage of your trial. If it's a multi-site trial, it'd be very unusual for you not to use clinic or sites. Often, if it's not that the sites are different in terms of how they treat disease, if you're dealing with patient subjects, at the very minimum, they might serve different populations. And those populations are defined by factors that are related to outcome. So usually clinics in there. In terms of other variables, I think looking to other trials, other published trials is a good idea. You can look at what maybe they use for their eligibility criteria, but that's not used for your eligibility criteria. That might be a good stratification factor. Factors where it's known that a treatment interaction exists, factors used by other trials, and factors that have been well known to be predictive of outcome. For instance, if I'm able to identify two important factors in my particular patient population, age and sex, what I might do is create these four strata that you see here. And this points out something that's really important, that if I'm dealing with a numeric variable, I have to categorize it. So of course, sex is naturally categorized, but or naturally categorical. With age, I'm going to have to come up with some kind of threshold ahead of time. So here I'm using 50 for the example, and that creates my four strata that I'm going to use. Now for each of these strata, I'm going to create a randomization list. Now I got to be careful how I create this randomization list, because just because I stratified them, it doesn't mean that's going to ensure balance. What I need to do within each of the stratum is use a restricted randomization procedure, most commonly block randomization. Because if I go back to this diagram, I kind of made it seem nice and neat that when I randomize these 6 people, I'd end up with 3 and 3 every time. That's not necessarily going to be the case, right? Unless I restrict it to be so, or at least approximately so. And so I'm going to want to use something like block randomization. And this is where the term stratified block randomization comes from, which is incredibly popular in the sciences. And that's going to limit the sort of imbalances that I'm going to see. I'm then going to implement the procedure, right? So as a subject comes through the office door, I consent them. And then what I'm going to do is I'm going to observe their age and sex. And if that person happens to be, say, a 65-year-old man, well, then they fall in the older male category. And this is the randomization list that I'm going to use. And so by doing this, what I'm going to end up with, and this is a great thing about this particular procedure, I'm going to have approximate sample size. And in terms of age and sex, I'm going to have approximately the same distribution. And so that's why this is such a popular procedure to use for randomization. One question might be, well, how many stratification factors can I use? And here you're going to have some limitations. You can't consider hundreds of stratification factors, even though you would love to do that, because you can end up with what we call over stratification, where you're going to end up with a lot of strata that are not filled or have minimum numbers. And so what I mean by that is, say that I consider two binary and two trinary factors. Okay, how many strata does that mean I have? 2 times 2 times 3 times 3, 36, right? And strata, remember, that's something we observe. So again, going back to my example here, I kind of acted like, oh, of course you're going to have 6 of each type. That's not the case, right? The reality is that you could have a pretty big imbalance, and you could have very few numbers in some of these stratum, and you could end up with some imbalances that you were looking to avoid in the first place. So what you need to do in that particular case, there's a few options. One is you could really reduce the number of stratification factors, or maybe use smaller block sizes. You might say, well, Greg, that's not enough. Well, how small is small? Well, what you could do is maybe consider some of these rules of thumb. I won't go over these, but this has been studied in the literature, the relationship between block size and the number of stratification factors. And these can provide a lot of guidance in terms of how you'd set up your trial in terms of randomization. In terms of analyses, a question might kind of get brought up here is, do I incorporate those into the analysis? And this was a big area of debate in the 70s and the 80s. Stat nerds like myself would argue about this sort of stuff. Do I just use these stratification factors in the randomization? Do I only do it in the analysis? Really, modern recommendations is to use both. If I'm using stratified randomization, that must mean, like in the example, my example here, age and sex, those must be factors that explain a lot of the variability in the data. It makes sense that I incorporate those into my analysis plan and do some kind of stratified analysis. There's also an advantage here of doing stratified randomization, is that helps a lot with subgroup analyses and tests for treatment heterogeneity. These are tests that are usually part of your trial, depending on the funding mechanism. Like if you go for a big PCORI grant, that's something they put a lot of emphasis on. And so having having a situation where you maximize your efficiency as much as possible, of course, is going to be really advantageous. In terms of other covariate adaptive randomization procedures you can consider, there is something called minimization. Why might I use minimization? Well, if I have a larger number of stratification factors. And so the basic idea of that is, after I randomize my individuals and the next person walks through the door, I'm going to create a score, which has this form down below here, which measures imbalance between my groups, right? I want to keep this mathematically simple. So let's kind of just go to an example and I'll show you how it works. It actually works pretty simply. So let's say that I have 60 people enrolled in my trial so far, and this is a summary of my age and my sex for those 60 people. So I see that I have 11 people over 50, 13 in group A. I have 13 over 50 in group B. I have 20 females in group A, 21 females in group B. And in walks the door number 61, right? And number 61 is a male of age 55. So he's male and he's in the higher age group. So what I'm going to do is I'm going to look at the data, and for those types of subjects, I'm going to see how my balance is. And so I'm going to create this next column here. And then for older people, older than 50, I'm 50 now, so I'm still in the young group, thankfully. So for those people, I have 11 minus 13, so I get a -2 here. So I can see that in terms of at least age with the older people, I've been favoring group B. And then with males, I've been favoring group A. Now, what I want to do is get this measure. What was on the previous page is this overall measure. And that's just going to be the sum of those two numbers. Now, the fact that this is negative, what's important here, is it 0, negative, or positive? The fact that it's negative, it means I've been favoring B in terms of those type of people. The next person gets A. Had that been positive, then that would have been the opposite conclusion. And had it been 0, that would imply balance. I'm going to randomly distribute to A or B using some kind of random number. And that's the idea of minimization. The issue here is that I'm going to have to update this information, and then I would move on to the next person. Now, there is a tweak of this. Some people don't like the idea, especially if you have an unblinded trial. that based on this data, I knew that the next person coming through the door, if they were an older male, I knew that they were going to get A. And so that would leave yourself open to sorts of biases that you might encounter in trials. Well, what I can do instead of being deterministic, I can be stochastic. And I can say, okay, well, instead of assigning them to A, I'm going to allocate them to A with just a higher probability. So it's not guaranteed they're going to get A, but they're going to get A with a probability of, say, I don't know, 2/3 or 3/4. And so that's known as a Simon-Pocock procedure, which is a tweak of what we saw. Now, the advantage here is I can accommodate many more stratification factors than I could with traditional stratified randomization. And this works really well if I don't have a big sample size. There are some disadvantages here. A big disadvantage is I cannot generate the randomization list in advance. And what I need to do is I need to keep continuous up-to-date records, right? Because my allocation was based on this table. It was based on the first 60 people that enrolled in my trial. And so that's a big disadvantage in the sense that you have to make sure that your trial is set up to accommodate that. Something like a multi-site trial might not be able to, you might not be able to do that. Another disadvantage that even though this will balance across the marginal distribution, so if I look at my two treatment groups at the end of the trial, they'll have approximately the same age and they'll have approximately the same sex distribution. But in terms of the cross classification, you could end up with an imbalance there just by the nature of how this procedure works. And then the fourth family of randomization procedures is something called response adaptive randomization. The idea of adaptive randomization is that if I start to see a benefit of one of my treatments, and really this has a place in the treatment of disease, if I'm dealing with a trial with patients that are maybe somewhat sick, right? Or very sick. So if I start to see during the trial that A is a superior treatment, it makes sense from an ethical standpoint. I'd want to give more people A as I go through the trial. And that's the idea of response adaptive randomization. Right? Now, for me to implement responsive adaptive randomization, of course, it needs to be feasible for me to identify what the best treatment is based on this outcome. And of course, you shouldn't do a trial if that's not true. So that's kind of a no-brainer. One thing I'd be a little bit careful of is that, I'm going to be making this decision based on the primary outcome. And the primary outcome is usually some kind of measure of efficacy. If the better treatment in terms of efficacy is also worse in terms of toxicity, this is not a good idea. This is not a good randomization to utilize. And then also the third thing I got to be careful of is I need to have a short-term outcome. I can't use long-term outcomes here. And so why might that be? Well, remember what you're trying to do here is you're trying to adjust the allocation to A, right? Based on the observed responses. If I have a long-term outcome and maybe a quickly accruing trial, I might be done accruing subjects by the time I observe the outcome corresponding to the first patient. If that's your case, this is a stupid procedure to use. You would never want to use this. This doesn't make sense. You want to use this with short-term outcomes. I won't go into the details of this because I'll be honest, the math gets a little bit thick with response adaptive randomization. There's things called play the winner where you continue to give patients the beneficial, what appears to be the beneficial treatment. Um, there's also urn-based designs. You can imagine that because we've talked about urns a few times now, that as I start to see the benefit of a particular treatment, I put more marbles of that type in that hypothetical urn. And then there's designs based on optimal allocation. Um, Now there's some disadvantages here. Your investigator, again, if you're not blinded to response, your investigator is going to have a good sense of what that next assignment might be. Again, I said this is limited to only short-term outcomes. The assignment list cannot be generated in advance, and that could create logistic problems. And then the analyses are more complex. The analyses are a little bit funky here, right? In the sense that if you think about it, if A is superior over B, then what should I see at the end of the trial? I should see an imbalance in the sample size, right? If A is looking beneficial, I'm going to be randomizing more people to A. If A and B are the same in terms of how well they perform, I'll have a balanced sample size. So your testing procedure is actually based on your observed sample size, right? Now that works okay for the primary outcome, right? Because I understand why that works that way, right? That's not an issue. The issue is with secondary outcomes, right? Your secondary outcomes, almost all trials have them. You might have some really important secondary outcomes. Because of how you allocated, you can't use traditional methods with the secondary outcomes. And that could be a big deal for you, depending on the importance of those secondary outcomes. Generally speaking, I've used this a few times, but really, if I'm worried about one of my treatments being superior over the other and me, maybe wanting to help patients as I observe that, I'm going to use something more like a group sequential approach where I analyze the data at different points during the trial. And if I see A superior to B, then I'm going to shut down the trial. And that will be the subject of a workshop I'll be giving in the spring on adaptive designs. So starting to wrap it up here in terms of conclusions, we've talked about a bunch of randomization procedures. I really covered all the popular ones. I'm not saying there's not other randomization procedures out there that might appeal to you, but this should really be it for 99% of you. You got to be really careful in terms of your selection. It's really easy to not think about this and really regret it later on, right? It's easy to say, oh, I'll just do the coin flip. I'll do complete randomization. And then later on, you see this really big sample size imbalance. You see this imbalance in terms of important covariates. It's too late then, right? You can't go back in time and magically create this balance. So this is something you got to give a lot of thought to during the planning of the study. And so with this sort of stuff, if you're having these sort of questions, what is the most appropriate randomization procedure for you? Really reach out to a statistician, right? I think, especially in trials, it's really important that you have a biostatistical collaborator. And looking for you at UB, you have some sources there. These are two different units that I direct here at UB. The first one is the BERD core, which stands for Biostat Epidemiology and Research Design, and that's located within our CTSI. That focuses a lot on early phase unfunded research. So for those that have pilot clinical trials where you want to, maybe assess the feasibility with the idea of getting some data that might lead to an R01 submission or maybe a big PCORI submission, that's stuff that we can help you with. We also do a lot with education and training and pathway development. If you're interested in requesting services, there's the website down below. Don't feel obligated to write that down. I'm happy to make my slides available later on, but that's really easy to find anyways. Just Google Buffalo CTSI, and you'll see in the upper right of the web page, I believe, a little button there where you can request services from the BERD core. And then the other unit I direct is an Office of Clinical Trial Development and Implementation. This is a new office that we actually just opened up this summer. This is housed within the VPHS office. And there we're focused on larger trials, either single site or multi-site. We do a lot with infrastructure creation to conduct these large clinical trials. We do a lot with statistical support and data management, as Austin will talk about during the next talk today. And for that, you could just contact me at my email. Really, the portal on the left side goes to my email anyway. So these are just two different ways to get into my inbox here. I'm easy to find. I'm in the Department of Biostatistics. Here are all the references if you're interested. And that's all I have for today. So I'll take any questions that you have. I just realized how dark it got in my room here. And of course, if anyone thinks of any questions, they can always email me later.
Okay, thank you very much, Dr. Wilding. Excellent. I want to welcome now everyone back to BERD 3 and clinical trials. And I want to welcome Austin Glick, who is a clinical data manager for the Office of Clinical Trial Development and Implementation. And he's going to talk about clinical trial data management. One, we needed to adjust the schedule a little bit because of an emergency, and he has stepped in and will be giving the second part of this talk next week. Thank you very much, Austin.
All right, thank you. All right, so hello everybody. My name is Austin Glick. So I am a clinical data manager. So today we're going to talk a little bit about clinical trial data management. Just give some background on me before we get started. I've worked in the field for many years now. I started out as a study coordinator working in oncology, moved into clinical data management, have been there for a while, recently working in industry before coming to UB, and now working in the Office of Clinical Trial Development and Implementation. So all of my experience is at a generally high level, doing very large clinical trials, lots of participants, things like that. So I'll be talking about kind of what a large trial would look like, what are some of the regulatory aspects of some larger trials, but also trying to transition that into what maybe would occur within data management in a smaller trial. All right, so first we'll go through the learning objectives. So the two objectives I have for you to take away from this presentation today is to identify the role and responsibilities of the clinical data manager throughout the different stages of protocol development, and then to learn about the many clinical data management tools and techniques used by the data manager and link those tools and techniques to the different tasks required in the protocol lifecycle. So I'm going to go through kind of the day, what a day of a data manager is, what they can do, how things change and differ between studies, and no study is the same and no tasks for a clinical data manager are the same. But I'm going to talk about the broad range of what that might entail, and then how the field has been shifting and how regulatory things have affected the field and how we move forward. So we're going to start by just giving an overview of what clinical data management is. So what is clinical data management? And this is a definition that I found that I like. There's not one general definition that everybody uses, but clinical data management is a critical phase in clinical research, which leads to the generation of high quality, reliable, and statistically sound data from clinical trials. So that's the important little tidbit right there. Is our goal as a data manager is to generate this high quality, reliable, statistically sound data. So everything we're doing is trying to get to that point and making sure that we're doing everything we can to get there. Clinical data managers are actively involved in all phases of the clinical trial lifecycle. Soon we'll be going through kind of those phases and what it looks like for a data manager, what they're going to be doing, and how those tasks evolve. And then some various procedures in a CDM include case report form creation. You'll hear me talk about CRFs as they're referred to a lot in this presentation, and then validation of those CRFs. Database design, so where we store the data, where it goes from the case report forms into our database. Quality assurance and quality control, also known as QA/QC. Data validation, so how we validate that the data was recorded and collected properly. Discrepancy management, what do we do when a mistake is made? Because that is a large part of the job. Medical coding, which is something that is done in a lot of modern day clinical trials so that we're being consistent. And that's something that will pop up a lot during the presentation. Data extraction, and then database finalization and locking. So what are the goals of clinical data management? So I split this into two columns here because it's kind of challenging to say, what are our real goals? Well, there's goals for a perfect trial, and then there's goals for where we realistically end up in data management. So in a perfect trial, we want perfect patient enrollment. We want no protocol deviations. We want no missed visits. We want no late data, no data discrepancies, no serious adverse events or SAEs. We want all of our visits to be completed per protocol and all of our specimens to be collected. Now that would be perfect. That would be an ideal study that would go through smoothly. We'd have no issues. But if trials were like that, I probably wouldn't have a job. So what do we do in the case of realistic goals? What are our goals when we're dealing with what actually happens in a trial? So we want to rapidly respond to enrollment errors. It's going to happen, and it happens all the time. Maybe something is done correctly, a participant doesn't meet the inclusion-exclusion criteria, they're inadvertently enrolled early, something like that. So we want to make sure that we're responding to those enrollment errors quickly as possible, and then we're adjusting our accrual goals, or we're taking them off study, or anything like that as fast as possible. We want to efficiently have a workflow in place for protocol deviations. Unfortunately, protocol deviations are going to occur. Most studies just, there's a lot of intricacies in a clinical trial. A site might make a mistake or something might happen, and that's okay. We just want to make sure that there's a workflow in place for reporting those properly, navigating how to give the site advice on how to fix it, and communicating with the protocol study team. And then missed visits under the protocol limit, we just want to make sure that we're monitoring those. If a protocol states how many visits could be missed before the participant is supposed to be removed from the protocol, we want to be monitoring that. We usually have some reports that are in place, or we're tracking that along the way. And then we're communicating with the site personnel to make sure that if a participant does hit that limit, that they know and they're aware that they need to take them off the protocol. We want to make sure data is keyed in a timely fashion. And that isn't just a kind of black and white thing here. We don't have a set time frame. It's always protocol to protocol. I'll use a phrase a lot, risk-based approach to data management. And with that, we adjust the keying timeline dependent on the data. If it is a serious adverse event, we generally want that keyed within 24 or 48 hours so that the protocol team knows if it needs to be reported to the FDA, it's reported to the FDA in a timely fashion, and the protocol team can figure out how to adjudicate that AE. But things like maybe medical history when a participant is in screening and it's a long screening period, that might not need a 48-hour keying time. So we'll give them a site 2 weeks, 3 weeks maybe to key that in there. So it's adjusting those realistic timelines and giving our site personnel enough time to make sure it's done, but also staying on top of it so that we're getting data flow in a good fashion. Addressing data discrepancies and correcting them in an efficient time frame. So these are data discrepancies, could be something as minor as just a falsely reported value with participant, they report their weight as 1,000 pounds. Obviously that's could be incorrect. So we query them, we go back and forth. Oh, it's supposed to be 100. They just added an extra zero in there. Great. They fix it right away. Quick and easy. But it could also be something like a date of birth was reported incorrectly, and we have to go through a longer data correction process. But we want to make sure we're doing that in an efficient time frame so that we get the data in the cleanest point as possible. So if we ever do have to pull any reports, they're accurate. We want to make sure SAEs are reported in a timely manner. That kind of goes under that data keying. But there's FDA requirements for SAE reporting. Most protocol teams want to know when an SAE is reported. So we're setting up things, maybe programmable items in the electronic data capture system so that when an SAE is reported, an email blast goes out and tells the entire protocol team what happened. Things like that, where we're pulling reports on a regular basis and adjudicating those AEs. So those are something that we're trying to keep ahead of. And then all data errors are addressed and logged correctly. There's a regulatory component called 21 CFR Part 11, which I'll talk about in a little while. But we want to make sure that we have everything logged, we have the correct audit trails in place, and everything is documented properly so that if we ever do get audited, which is a big part of clinical data management, that we're prepared and we're ready. So what is a typical day for a data manager? A typical day could include any of these tasks, not necessarily doing all of them, but could be completing data review, could be participating in team meetings with the clinical research teams, could be generating and reviewing reports, could be addressing any outstanding site questions or queries within the database, could be communicating with the site personnel to provide updates and training on the study, completing any required data correction tasks. Obviously, those are ad hoc, but they take a bit of time to do, and then documenting all data management activities and maintaining accurate records for audit purposes. A data manager isn't generally doing the same thing every day. We're not reviewing data every single day. We're not running reports every day. Our jobs and our week to week varies so greatly based on the protocols, what life cycle they're in, what's required for them. If we have review schedules, do reviews fall on the same days, things like that. So the field shifts and evolves, and the job shifts and evolves, and that's why it's very interesting. So what are some ad hoc tasks for a data manager? This could be case report form creation. We have a new study and we need to create a whole new set of case report forms, or maybe we have a new form that's required. There's an amendment to a protocol. They want to add a new questionnaire, and we got to get it into the database so that the sites can fill it out. So we have to build one of those, and then we need to test and validate those forms and/or the database. So that could be another ad hoc test. These are things that happen all the time, but they do happen frequently enough that we have to be prepared for them. Preparation of data sets for analysis, if there's an interim analysis or a final analysis, we're doing a lot of data cleaning. We're making sure all the data is correctly, put away in the database properly. And we're doing a lot of preparation for those analyses. So that could be something that pops up. Building or maintaining the randomization system. Data managers generally are heavily involved in the randomization system. It's usually integrated in an ideal case into the electronic data capture system. So all of our technologies in one place. So we're dealing with that. We're maintaining it. We're making any changes if a protocol changes and the IE criteria are different now, things like that. Creating or maintaining views, tables, and reports used for data review. Our jobs are for reviewing data can be really challenging. You can have a study that has 2,000 participants, and each participant has 5 visits, and 20 forms at each visit, something like that. And we have then an immense amount of data to review. And we can't physically, as one person or it's even a team, review all that without going insane. So we create views and tables and reports that we use to help us automatically review that data. So we're getting outputs, we're putting in some things in there that are like highlighting any at-risk data, things like that, so that we can take a deeper dive. It's very difficult to just scroll through data for hours on the day and try to find issues. It doesn't work that well. So we're always trying to improve the process. Doing regulatory compliance checks. Like I said previously, audits are a big risk, and audits are something that data managers are always thinking about. Because at the end of the day, if an auditor comes and it's a government audit, we're the ones that are communicating with them a lot, and a lot of our day-to-day tasks are being heavily scrutinized. So we're completing regulatory compliance checks. We're making sure that everything is up to date. We're making sure that we're following our procedures that we have in place, so that if we do have an audit, it goes smoothly. And then we're doing process improvement tasks, working on working groups, doing SOP writing or updates, continuing education. The field is constantly evolving as technology evolves, data management evolves. And so it's always about learning, it's always about improving, and it's always about trying to make the next big leap to having the highest quality data. So next we'll kind of jump into a really important thing when it comes to data management, and that's what our workflow looks like. How do we move through the evolution of a study, and what do the day-to-day tasks kind of look like with each phase of a protocol? So this is a nice chart. We're going to come back to it. We're going to go between the different phases and break everything up and go through it in a nice orderly manner. But this is the data management workflow at a very high level. There's lots of individual tasks peppered in here in between different phases, lots of things that are going on. But this is a very simple view of what a workflow might look like for a data manager. So we're going to start right here at the beginning of protocol review and development. The data management workflow starts during protocol review and development, like all other parts of clinical trial. Data manager during this step assists the study team during the development process and weighs in on any data-related questions or topics of discussion. And the DM will begin to plan out what data management tools and CRFs will need to be created based on the developing protocol and needs of the study team. So this is one of the lighter steps for data manager. We're going to sit on those calls and we'll be involved in some of the discussions. We'll be talking about kind of data capture systems. Do we want to use something like RedCap? Do we want to use Encore? Anything like that? But this is a lighter phase for us. But then we jump into some of the setup phase, and this is a really important part that kind of sets the pace for the entire trial. So for data collection needs, one of the most important steps during your early protocol development is identifying what types of data collection elements need to be included in the study. This could include case report forms, either electronic or paper. Sometimes paper are used, a randomization system, any reports, sponsor level or study team level, setup of an electronic patient-reported outcome system, also known as ePro, could be called, they're just electronic questionnaires, could be done on a tablet or a cell phone, things like that, so using more technology. Then the next step is to identify what electronic data capture system will be utilized for the study. There's a wide range of EDC systems available, and each has its strengths and weaknesses. A lot of smaller local studies can be built in REDCap. REDCap is free, it's great, it's wonderful if you have a smaller trial that's going on just at a single site, but it raises some challenges for use on larger multi-center protocols. So for these studies, the data manager will work with the team to discuss the options available. Systems like Encore, Open Clinica, Metadata Rave, and VDoc, to name a few, are some big, larger EDC systems that have a little bit more technologically advanced, have a lot more features, but they're also a little bit more costly. And then DM will go through the benefits of these systems, such as the capabilities, ease of use, reporting capabilities, and available add-on utilities, such as randomization. And then they'll discuss the regulatory considerations for each system. So most cloud-based EDCs are 21 CFR Part 11 compliant inherently. But a system like REDCap needs to be configured locally to be compliant. So as a consideration that needs to be made when thinking about this, because you have to put a little bit more extra steps in the beginning to make sure you're compliant on the regulatory end. And then based on the EDC system selected, we would then identify with the team what additional utilities are available, and from those, what the team would like to use. So for example, if a study is multi-center and has a complex randomization structure, the data manager, along with the statistician, could present on implementing an online randomization system or using an EDC system that has that built in, because it's going to save a lot of hassle in the future. So then the next part, after we decide what the EDC is used, what kind of things we need to do, and what utilities we need to create, we have to make the CRFs and set up the database. So once the EDC is identified, the next largest step is developing the electronic case report forms, or ECRFs. This step will vary greatly based on the study. The data manager is going to take the protocol and develop a dynamic, rigorous set of forms that will follow good clinical data management practices, something I'll touch on a little bit in the future, but it's just a guideline for how to do good data management. And these forms may also use the data standards set by the Clinical Data Interchange Standards Consortium, or CDISC. And CDISC is an organization that tries to make data and CRFs friendly and be consistent across studies. So I'll talk about this a little bit in the future, but these standards kind of lead to a cleaned final CRF and its data that's organized in a predictable format. And then based on the CRFs that are created, a database structure needs to be defined and data mappings need to be set up. So like with the ECRFs, this can vary greatly from study to study. Smaller studies will require less complicated structures to be put in place. In contrast, larger studies that require submission to the FDA will need SDTM and analysis data model, or ADAM data sets, to be set up. And this requires more work to put in, and you need to make sure that it's set up properly. But most systems will be then set up in a SQL or SQL database to ensure data integrity and safety. And then on the right side of this slide right here, that is an ECRF. It might be a little bit small, but it's just kind of what a CRF looks like, what an electronic form would look like on here. So, we're vital signs performed. This is just a vital signs form. They would select these options, they'd fill it out, they'd submit it, and then it would go and translate into the database. So then the next step before we move to study opening is user acceptance testing and validation. So data managers do a lot of testing. Testing and validation is going to occur for each data element created during the study build process, and also after that process if any changes are made. We have big, rigorous testing processes set in place. We need to make sure everything's tested properly. We need to make sure everything's good to go. So we're constantly testing things, and that's to keep us audit ready, and that's to meet all of our regulatory compliance requirements. So ECRS will undergo intense testing and validation to ensure that no mistakes were made during the build process and that all forms flow properly. Love to say that we catch all mistakes during testing, but sometimes that doesn't happen. But we're just trying to test them. We do things like chaos testing, where we try to break the form as much as possible to see if we can get it to fail in some way, or see if we can find something that doesn't necessarily make sense when you're reporting data. so that we can fix it before we actually have, it at the sites, and the sites are keying data and putting participant data in there when it's a little bit more challenging to change. And those testing teams will consist of individuals with a broad range of expertise and experience to ensure that all data elements are designed per our best practices. We have people who are our site coordinators that might come in and join us. Our statisticians might be involved to make sure that the data flows properly. Other data managers from other groups, maybe. We just want to have as many hands in there to review that as possible to make sure that we catch any and all errors. And then the validation process will consist of sign-offs from both the build team and the study PIs, make sure that we have all of that documentation in place to show that we did properly test everything. And that brings us to the longest part of the kind of study life cycle is after study opening. What do we do? How does data review occur and things like that? So once all the necessary data systems are in place and validated, the study can open and start accruing. And while the study is open, data review and validation will consistently occur to ensure data integrity. These processes are going to vary based on the complexity of the study, like all things, but will be defined in the data management plan, or the DMP as it's called. One element of data review and validation is source data verification, also referred to as SDV. This may or may not be done by the data management team based on the study. Larger multi-center studies typically utilize study monitors or clinical research associates, as they're called, CRAs, that perform a targeted source data verification, also known as TSDV, and act as an independent entity. Studies where the data is collected at UB could leverage UB personnel to perform source data verification. And source data verification is really just taking a look at the source documentation, maybe a patient chart, maybe a paper form that's completed, a questionnaire that's completed on paper, and just comparing it to what's in the database so that we know that things are reported properly. The biggest hangup as a data manager is I personally don't have access to the source data, and that's by design. I should be kind of a step away from the participant side of things, the clinical side of things, but that makes it more challenging for me and other data managers to verify and validate that the data is reported properly. If there's a decimal place off on a lab value, I don't have that lab report, so I can't verify that myself. So that's why we rely on source data verification. And this kind of helps identify issues and set up future data review processes, which is really important. And then we do data querying, and that's based on our data review, and it's going to occur consistently through the life cycle of the study. Data managers are always going to be querying data. We're going to be working with the site personnel, the site data managers or study coordinators, going back and forth and kind of working on fixing issues that might pop up. So the DM and the data manager assistant as well will utilize data review best practices and integrate a risk-based approach to data review to ensure the highest quality data is presented in the final data sets. And then they're also going to set up reports. So those are going to be run to present the study team with insights into the study's data integrity and, current accrual rates, what's going on, and if anything needs to be done. And the emphasis on that risk-based approach, like I said, we can't review everything under the sun. So we develop a risk model for what data needs to be reviewed more intensely, how often it needs to be reviewed, what the timelines for that might look, and how we create reports for those things. So something like an AE log is going to be reviewed a lot more frequently than maybe a minor questionnaire that is just done once per the study. And then data issues that are identified for significant data points, this could be like participant ID, date of birth, and treatment arm, will be corrected using a set data correction process. This process will allow for proper documentation of important changes and can be presented to key personnel in case of an audit. So data corrections may not need to occur for a study. A study might go through its entire life cycle, never need a data correction because all of these key points were always entered correctly. But if it does need to occur, we need to make sure that process is set up because this is something that auditors look at. I've been through many government audits in my career, and they're always asking about data corrections. How many were done, what was corrected, how fast was it corrected. They're looking at our documentation. They're making sure the database looks okay because these could be significant issues and they could really, really affect the overall quality of data. And then we go to the last little phase here, and that's database lock and study analysis. So for database lock, once the accrual goal has been reached or a study is prematurely closed, we close the database. So this is a process, though. First, the data is going to undergo a thorough review and cleaning. All outstanding data queries will be addressed, and data validation will occur. We're also going to freeze accrual. So accrual is not going to occur anymore. We're going to freeze entry of new data at one point once we're sure that all the data has been entered. And then we're going to go through with a fine-tooth comb and we're going to review a lot of the data. We're going to make sure everything looks correct. We're going to ask the sites to review all the data. Anyone who's entering data, want them to review to make sure they didn't make any mistakes. And then once we can say that everything is sufficiently clean and no issues remain, then we will lock the database for data entry and that'll be done. We'll sign off on it and everything will be set. If we freeze it, we can always unfreeze it to make any corrections. So there is a little bit of a stopgap in there while PIs kind of sign off on the data. But once we lock it, we're not supposed to unlock the data. It says that it's done and it's ready for analysis. And then once that happens, analysis can occur, and they're going to use pre-established data sets that we're hoping we set up in the beginning of the study. You know, if we're using SDTM or ADAM, that's going to be developed in the beginning or throughout the life cycle so that we're ready for this final phase. All right, and so that's the entire data management workflow, you know, from the start all the way through kind of the study analysis. And a lot of extra things are going on in these phases. A lot of this stuff occurs, you know, this is a long period of time too. Some studies might have long accrual periods, 5, 6, 7 years. And so we're sitting in that middle portion for a really long time, but it tends to be a mad dash to get a study open and ready. And then, you know, we're cruising out. It's an endurance race as we're keeping good data practices, and then a mad dash to get the study analyzed and locked. So we're always prepping. We're always trying to stay ahead of this process. All right, so then we're going to talk about briefly, because you can't talk about data management without talking about some regulatory considerations, but I promise, I know it's almost 5:30. I'm not going to talk about too much regulatory stuff. It's 3 quick slides. We'll go through the 3 important ones that everybody should know about when it comes to data management, and then we'll move on from there. So the first and arguably the most important regulatory component that data managers deal with on a daily basis, and I mentioned it before, is 21 CFR Part 11. You can see that the front page of it over there. It was written in August of 2003. It is issued by the FDA, and it sets the requirements for electronic records and electronic signatures. And why is that important is because it establishes the criteria for creation, modification, maintenance, and transmission of electronic records, and it mandates controls to ensure the integrity, confidentiality, and availability of electronic records. So this is overseeing all electronic capture. And when this was put into place back in 2003, electronic data capture wasn't as big of a thing. But nowadays it is compulsory. So all data is primarily being captured on electronic means. Some studies still will use paper forms that are entered in, but a lot of them are moving straight data entry into an EDC system that goes straight into the database, and there is no paper involved in it unless a site coordinator wants to print out some paper CRFs for in-clinic. So this component really, really is important, and it regulates how we build our EDC system, how we track things, how we maintain things. And it really is there to generate secure computer-generated time-stamped data. And part of that is generating these audit trails that I think I've mentioned briefly here for electronic records. When you're using an electronic data capture system and you are audited, one of the first things that they will ask you for is an audit trail from your EDC system. It needs to be 21 CFR compliant, and the actual regulations say that it should be immediately there, ready to be downloadable as a PDF format and given to an auditor on the spot. So that is a big regulation, and that's why big cloud-based EDC systems put a lot of work into making sure that their systems are ready to do that from the get-go. It doesn't matter what we do to the system, it's ready for that immediately. And these auditors are going to be looking for that, and they are going to ask for it. So it's important to think about. And then systems must be validated to ensure accuracy, reliability, and consistent intended performance. So it's just overseeing how we collect data, how we do things. And this has been kind of grouped into best practices, but this is incredibly important for what data managers do. And then the next thing is important for all clinical trials, and that's ICH, good clinical practice. So this is something a lot of people who've worked clinical trials are familiar with. It ensures the protection of trial participants' rights, safety, and well-being, establishes standards for designing, conducting, recording, and reporting clinical trials to ensure data integrity and reliability. It specifies requirements for data collection, management, and storage to ensure accuracy and confidentiality. And it outlines the procedures for reporting adverse events and ensuring participant safety throughout the trial. So from a clinical trial standpoint, very important regulation. From a data management standpoint, it tells us how we should be collecting data, how we make it secure and confidential, making sure that there's security things in place, making sure we're only using trusted systems or storing it properly, things like that. Nobody has access to data that they shouldn't have access to. Like I said a little while ago, data managers are kind of a step away from clinical staff. There's a lot of things that we shouldn't be exposed to. I should never see a participant's name. I shouldn't see like their address or telephone numbers. We want to keep that data separate so that we're maintaining confidentiality and we have that buffer area. Not saying the data manager can't access that data, but it's good practice to try to stay away from it. And then adverse event reporting is really, really important. And it's a lot of what data managers have to do kind of ad hoc, make sure that we're maintaining the AE logs, the AEs are reported properly. If it's an SAE, it's reported to the study team properly because it's going through our database. So we're ensuring that we're following GCP. And then the last one here for regulatory considerations is our FDA reporting requirements. So there's a couple of reports here that data managers have to concern themselves with. So safety reporting, any serious and unexpected adverse events need to be reported to the FDA within 15 calendar days. This is specifically referring to a study with an IND. So obviously not all clinical trials. This is, if you're doing an IND study, you need to report this, but we're making sure that we have that in place. And it's also best practices to make sure that serious adverse events are being reported properly, and we have mechanisms in place to get that information out as quickly as possible once it's reported. There are annual reports that progress reports on the clinical trial that include updates on patient enrollment and any significant changes if they're required. So these are kind of something that we help design and help develop along with the statistical team. So data managers are involved in that process. A final comprehensive report upon the completion and termination of the trial is required. Again, something that we help pull some data for and help develop. Protocol amendments, so any significant changes to the trial protocol are required to be reported to the FDA. Not necessarily something that we're doing as data managers, but something that we're kind of aware of, and protocol amendments are something that affect us greatly. And then investigator reports, and investigators must submit these reports on the progress of the trial and any adverse events. Again, something that an investigator is in charge of doing, but they might come to the data management team and ask for help with that, or ask us to pull some data. So it's something that we need to be aware of. All right, so then we move on to some guidance and standards. The field has evolved greatly over the past 20 years. There's been a lot of new guidance, a lot of different standards, a lot of changing in how we do certain things to keep our data quality better as we strive to be better. So these are some of the things that we specifically use. So the first is good clinical data management practice. Kind of mentioned this briefly. And this is a group that creates this, that does specifically data management. They're not government agency, they're a nonprofit. And they develop this basically guidebook for data management. And it goes through everything. It ensures data accuracy, completeness, and reliability through robust validation techniques and regular data reviews. So it gives instructions on how to do all those things. It emphasizes compliance with global data protection regulations, and the emphasis there is on global. I'm talking about a lot of U.S. standards, but if you have a trial that is international, following those global protections is also really important. So GCDMP does address that. It promotes the use of standardized methods and electronic case report forms to ensure consistency and comparability across studies, something that's constantly harped on. How do we make better CRFs? It encourages regular updates and real-time monitoring to prevent delays and ensure timely decision making. How do we monitor? How do we make sure that data is accurate? And then it takes a risk-based approach to data management, focusing resources on critical areas by identifying and mitigating potential risks early in the trial. And this is relatively new. Not a lot of data management still follows this risk-based approach yet. They're still doing it the old-fashioned way of trying to review 100% of all data. But risk-based approach is kind of the future of the field. How do we develop early on ways and methodologies to help us in the future? We identify high-risk data points and we focus a lot of the clinical data management on that. And then all the other things still get reviewed and we still have processes in place to protect that data, but it takes more of a backseat so we can focus on the really important data and the really important aspects of a clinical trial. The next one is the Clinical Data Interchange Standards Consortium, or CDISC. This creates the Clinical Data Acquisition Standards Harmonization, also known as CDASH. Data management is a lot of acronyms, constant acronyms, unfortunately. But CDASH establishes a standard way to collect data consistently across studies and sponsors that data collection formats and structures provide clear traceability of submission data into the study data tabulation model, or SDTM, delivering more transparency to regulators and others who conduct data review. Essentially, this is a fancy way of saying that this sets a model for how to collect data. And if you follow that, data is consistent across protocols. The end of the day, the goal is that if you give that data to some other researcher from a different institution and they take a look at it, they understand what's going on because you're following a standard. And we're trying to follow a standard so that we can do that and that research is communicable between organizations. So we implement these so that does happen. And then SDTM leads into the analysis data model, which is more focused on the statistical side, which is an efficient generation, replication, and review of clinical trial statistical analyses and traceability amongst analysis results, analysis data, and data represented in the SDTM. So ADAM is more of a way to translate SDTM into an analysis data set. So speaking of SDTM and ADAM, just giving you a comparison here because these are important. If you have a study that needs to report to the FDA, they actually require SDTM and ADAM data sets to be reported. So they're very important to understand. So for SDTM, the purpose is to standardize the format for organizing and submitting clinical trial data to regulatory authorities. The structure divides data into domains such as demographics, DM, adverse events, AE, and laboratory results. Those like two little letter acronyms are the standards for the domains themselves. So you'll see those pop up in SDTM datasets. The data collection captures raw data collected during the clinical trial. So this is where the raw data goes. Once it's captured in the EDC, it funnels into the SDTM domains, as they're called. And then that data is where that is stored. And then for regulatory compliance, ensures the data is consistent and compliant with regulatory requirements. ADAM, in contrast, facilitates the creation of data sets for statistical analysis and reporting. It's structured to derive from SDTM data sets, and it includes both raw and derived data. So it's not just raw data in ADAM, it's pulling, in the life cycle of it, it's going from EDC to SDTM, and then it's pulling SDTM data directly in. It's also deriving some data from SDTM. The traceability ensures that clear lineage from raw data to analysis results. And then it's designed to support efficient generation and review of statistical analyses. So ADAM is there for the same reason SDTM is there. It's a controlled way of analyzing data, and it's really important for a lot of these things. And then the last thing here that I'm just going to touch on briefly is medical coding. So some medical coding products are out there, and medical coding is something that's done a lot in clinical data management. A lot of clinical trials are moving to medical coding. I just have two on here. There's a ton of them, but two that I wanted to address is Common Terminology Criteria for Adverse Events, or CTCAE. And it's developed and maintained by the National Cancer Institute, and it is a descriptive terminology which can be utilized for adverse event reporting. I only narrowed out this one because I've used it a lot in the past, but there are a lot of AE reporting criteria documents out there. for different NIH-funded groups. A lot of them have their own things. CTCAE is probably the biggest, but a lot of them have their own, and you'll work with these a lot with clinical trials that are NIH funded. And they specify what an AE is, how to grade it for the sites so that they're not grading it based on the provider's opinion. It is more of a set guidance for how to grade AEs. And these can be incredibly complicated, but they are coded in a way that allows us to be consistent. So, some of these AEs have codes associated with them, and then we can link that code in the database. And then if somebody else, again, were to pull the data and look at it without knowing how the trial was conducted, they could see those terminologies, they could look up that code, and they can get exactly what they're looking for. And then the other one, which is really important, is MEDRA, which is Medical Dictionary for Regulatory Activities. And this is a rich and highly specific standardized medical terminology to facilitate sharing of regulatory information internationally for medical products used by humans. Essentially, this is a big dictionary that can be used to code any types of data. So they have things in there for drugs, pharmaceuticals. They have some adverse event stuff in there, patient visit things, laboratory values. There's a lot of codes within Medra that we want to link. And linking those codes, again, allows for traceability of that data. How did it flow, and how information can be shared internationally, specifically with other researchers, and they know how our studies were conducted. All right. Well, that was the whole presentation. So I guess I'll open it now for any questions.